Hi everyone, and welcome to our video on students' t-test. This is part of our Module 4 Part 3 series to do with classification and evolution, and in this video we're going to be having a look at the specification reference M1.9, so one of the maths references. Select and use a statistical test. So the statistical test we're going to be looking at in this video is students' t-test. In terms of when we would use a t-test, this would be when we're comparing two means. And basically what it's going to do is it's going to tell us if the difference between two means is significant or not. There are two types of t-test that we could use: paired or unpaired t-test. So a paired t-test is what we would use when the data points are related in those two sets of data. For example, it might be before and after readings in an experiment. The unpaired t-test is when those data points are unrelated. So you might be looking at masses of two separate populations of rabbits, for example. The first thing that we're going to do with our t-test is we need to understand this thing called the null hypothesis. So the null hypothesis is just that there is no significant difference between the means of the two data sets. And that's the same whether it's paired or unpaired. We're just stating that if it's the null hypothesis that is correct, there is no significant difference between those two means. What we then need to do is obviously select the right formula. And I say select, but on your exam paper, if they want you to calculate the t-test, they just print it for you. You don't have to memorize these. They will be given to you in the question that you are going to use it in. And the people that write your A-level biology papers are not mean enough just to give you a whole formula sheet you've got to select from. They're not going to give you the two t-test formulas and expect you to work out which one to use. If they want you to use it, they will literally just print the formula you need. So, unpaired t-test on the left, paired t-test on the right. The key thing that we've got to know though, just like all of these stats test formulas, is they're not telling you what any of these bits mean, okay? So you do need to know what the different sections stand for, just so that when they give you the formula, you know what you're sticking in where basically. So if we look at unpaired first of all, we've got on that top line x1 and x2. So x1 is the mean of sample 1, x2 is the mean of sample 2. Then we've got S1 and S2. So S is our standard deviation, obviously of sample 1 and sample 2. And then we've got N1 and N2, and N is just the sample size of, again, data set 1 and data set 2. The paired t-test, we've got our D bar, so that is the mean difference. N is our number of data sets, and our SD is the standard deviation. Now, what they're not going to do, as far as obviously the exam papers that you are going to get will be, they're not going to ask you to calculate the full thing from scratch, because that would mean you're having to calculate the standard deviation for two data sets before you can even start the t-test. That's going to take a massive chunk of your exam time. So it's going to be a massive weighting towards one type of skill, and it's going to deduct from the rest of the skills they can test you on, so it won't happen. They're just going to give you the standard deviation if they want you to calculate t-test. So I've just given you this little example here where we've got our two data sets. We've got 10 bits of data in each set because our n is 10. The mean for data set 1 is 86.3 and the standard deviation 7.45, whereas the mean for data set 2 is 84.6 and its standard deviation is 8.98. So what we're going to do is we are going to be using our t-test formula that I've given us here. And what we do is just substitute in those values. So x1 is 86.3, x2 84.6, so 86.3 minus 84.6 divided by the square root of. And make sure we're using brackets appropriately on our calculator here, folks, to not get weird answers. So standard deviation 1 squared, which is 7.45 squared, divided by 10, plus standard deviation 2 squared, 8.98 squared, divided by 10, and that gives us our answer of 0.46. So you need to practice with the calculator you are going to be using in your A-level biology papers well in advance of going into that exam hall. please don't be that person that asks the invigilators for a calculator on the day, because I guarantee you won't be confident in using it. And if you're not confident in using your calculator, you make stupid mistakes and throw away what should be pretty straightforward marks to get. The next thing we need to do is actually work out if this value we've now calculated is significant or not. So what we're going to do is compare that value we've calculated to a table of values. And in order to do this, we need something called the degrees of freedom. So to work out your degrees of freedom, all we need to do is the total sample size of all your data sets minus the number of data sets. So in our example that we looked at, we had 2 data sets with 10 bits of data in each. So our sample size is 20, and we divided up into those 2 sets. So 20 minus 2 gives us 18 degrees of freedom. You will then get given a little table or an excerpt from a table like this, where we've got different degrees of freedom, and the values they give you will all be potentially things you could have calculated using those data sets incorrectly, and of course the one correct one. And then we've got our value of t at the 5% significance level, typically. So what we then need to do is we need to use our calculated t value and then compare it to the correct row in our table to see if the difference is significant. So in our case, obviously, we had 18 degrees of freedom, so therefore we are looking at this row here. So our 5% significance level value that we're comparing to is 2.10. Now, our calculated t value, if you remember, was 0.46. And we're looking at, obviously, our 18 degrees of freedom, so we're comparing to 2.10. Now, all we've got to do is look to see, is our value greater than or less than the value in the table? So 0.46 is less than 2.10, therefore our difference is not significant. We would therefore accept the null hypothesis that the difference between these two means is not significant. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over into the website where you can find a range of other resources to help you in your A-level biology studies.